Assalamu alaikum guys and welcome to my YouTube channel Biology Articles and MCQs. Today we are going to start a topic that is glycolysis. Guys, glycolysis means splitting of sugar. In this topic, we will study that how the glycolysis or the sugar split into its component that is called pyruvate. Sugar means we know that the simplest sugar is glucose. So this process will start from glucose and ends after the 10 steps in the product that is called pyruvate. So when we need glucose or when we need energy, this process is carried out in our body and we split the glucose into the pyruvate and the energy that is in the form of ATP, adenosine triphosphate, and in this way this glycolysis will takes place. So before starting the process of glycolysis, we have to know that where the glycolysis takes place in our body. Here you are seeing a cell. There is the main outer membrane that is called cell membrane, a main portion that is called nucleus which contains the DNA, and the third portion is called cytoplasm which is a viscous material. You can say that it is a viscous material of the cell. So the glycolysis takes place in the cytoplasm. Means when we need glucose, the glucose present in the cytoplasm of our cells split or break down into its products, and that process is called glycolysis. So let's start the process. Here is the flow chart of the whole process of glycolysis. We will study in the video all these steps one by one in detail. The glycolysis process is starting from the glucose and it is ending at the pyruvate, and these are total 10 steps which we will study next. So starting from the first first step, that is the glucose will first change into glucose 6-phosphate. There is a glucose molecule, means it is the simplest sugar which we need to split. Here is a glucose molecule, and you know that the glucose is a 6-carbon molecule, and in the ring structure it is present in the six cornered ring structure. Here is this is the carbon number 1, then this is carbon number 2, carbon number 3, and then 4, carbon number 5, and here is the carbon number 6. You are seeing that the carbon number 6 have OH group is attaching to it, and that OH will replace with the phosphate group. How it will replace? It will replace by the splitting of ATP that is already present in the cell. The ATP, that is adenosine triphosphate, will split into ADP, that is adenosine diphosphate, and you know that the ATP has a total 3 phosphate molecules while the ADP have 2 phosphate molecules, means the ATP is now is converted into a 2-phosphate molecule structure, that is ADP, adenosine diphosphate, and the one inorganic phosphate will release. This one inorganic phosphate will attach to the carbon number 6 of glucose in replacement with this OH. Here this phosphate molecules that comes from the ATP is attached to the carbon number 6. In this way the glucose is converted into glucose 6-phosphate. Glucose 6-phosphate means that the phosphate molecule is attaching to the carbon number 6 of the glucose. Means I have already told you that this is the carbon number 6, and this phosphate is attaching to the carbon number 6 of glucose molecule. So this molecule is called glucose 6-phosphate, and this process is carried out by an enzyme that is called hexokinase. Hexo because this is a 6-carbon sugar. So this is the first process in which the glucose has converted into glucose 6-phosphate. In the second process, the glucose 6-phosphate will now convert it into fructose 6-phosphate. Here is the glucose 6-phosphate that the phosphate molecule is attaching to its 6 carbon, and it will convert it into fructose 6 phosphate. You know that the glucose and fructose are the isomers of each other. What does the isomer mean? The isomers means that the molecules that have the same molecular formula but different structural formula. The molecular formula of glucose and fructose is same, that they both contain 6 carbons, but here you can see that the structure of the glucose is a 6-corner ring structure, while the structure of fructose is 5-cornered ring structure. So they differ on the basis of their structure only. So the enzyme that is carrying out this process is phosphohexose isomerase. So this isomerase enzyme is converting glucose 6-phosphate into its isomer, that is fructose 6-phosphate. There is only the change of the structure, and the structure is changed from the 6-cornered ring structure into the 5-cornered ring structure. In the third step, that fructose 6-phosphate will now convert it into fructose 1,6-bisphosphate. Here you can see that fructose 6-phosphate has a phosphate group attaching into its 6 carbon. But now a second ATP is used. The first ATP is used in the first step, and now the second ATP is used. The ATP will convert it into ADP, and the one inorganic phosphate will release, and that one inorganic phosphate will then attach to the carbon number 1 of the fructose 6-phosphate, and now it will become fructose 1,6-bisphosphate. Means now this molecule has two phosphate groups, means bisphosphate means 2 phosphate groups, one attaching to its carbon number 1 and the second is attaching to its carbon number 6. So naming this molecule as fructose 1,6-bisphosphate. The other the other whole structure is the same as the fructose 6-phosphate, and this step is carried out by the enzyme phosphofructokinase 1, that is also called as PFK1. Moving toward the fourth step, where the fructose 1,6-bisphosphate now will split down into the two different molecules, and these two different molecules will contain 3 carbon atoms. Here there is splitting carried out, and the carbon number 1, 2 and carbon number 3 of fructose 1,6-bisphosphate will combine into a structure that is called dihydroxyacetone phosphate, and the other 3 carbons of fructose 1,6-bisphosphate, that is carbon number 4, 5 and 6, will change into a molecule that is called glyceraldehyde 3-phosphate. Means this molecule has the phosphate group attaching to its 3rd carbon. So this 6 carbon molecule is now converted into 2 different molecules, that these molecules are 3 carbon molecules, and this process is carried out by aldolase enzyme. In the next step, the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, means these two formed. These are also isomers of each other, means they both have phosphate molecules, and they both have 3 carbon atoms, and the number of oxygen and hydrogen atoms in these both molecules are also same. So these are also isomers. And in the 5th step, we are seeing that these are also interconvertible, that dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate, and the glyceraldehyde 3-phosphate can be again converted into dihydroxyacetone phosphate by the use of an enzyme, that is triose phosphate isomerase. Triose means these both molecules are containing 3 carbon atoms. So now the next step, before going to next step, we have to go through this flow chart, that we have reached this step, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now you have to learn this one thing, that glyceraldehyde 3-phosphate will convert it into further conversions as same as the dihydroxyacetone phosphate. Means one glyceraldehyde phosphate will convert into 1,3-bisphosphoglycerate. The dihydroxyacetone phosphate will also convert into this same thing. Means there will be the formation of two 1,3-bisphosphoglycerates, and then next there will be formation of two 3-phosphoglycerate, and the two molecules of all these molecules will now form. So in these steps there will be the interconversions of two molecules also, means two NAD used and then two ADP will be used. So moving toward the next step, here I have shown you glyceraldehyde 3-phosphate, and you know that the dihydroxyacetone phosphate, that is the isomer of the glyceraldehyde 3-phosphate, will also converted into 1,3-bisphosphoglycerate at the same time. One thing here you can see that first of all we see in the first and third step that the ATP was using. But now we don't have to use ATP, we have to produce ATP because it is an energy producing process. Glycolysis is an energy producing process. So for the conversion of glyceraldehyde 3-phosphate into its product, we have to now produce ATP. Means here is one inorganic phosphate that will be used, and that inorganic phosphate will attach to this carbon of, again means the carbon number 1 of glyceraldehyde 3-phosphate here this, and it will convert it into 1,3-bisphosphoglycerate. Means the carbon is attaching to the carbon number 1 and carbon number 3 of this molecules. The phosphate is attaching to the carbon number 1 and carbon number 3 of this molecule. At the same time, the NAD positive is also changing into NADH and hydrogen, because this hydrogen is removed, and this hydrogen from glyceraldehyde 3-phosphate combined to the NAD positive and converted into its product, that is NADH. So in this step, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Then 1,3-bisphosphoglycerate is again using an ADP. Means this ADP will now change into ATP as in the previous process. In the previous step we have just used inorganic phosphate, you can see. But now in the 7th process we are using ADP. The 1,3-bisphosphoglycerate is now donating its 1-phosphate molecules to ATP and ADP, and this ADP will then convert it into ATP. This ADP has already two molecules of phosphate. One is donated by now 1,3-bisphosphoglycerate and will change into the ATP, adenosine triphosphate. And the product will also a 3-phosphoglycerate. Means now this molecule only have one phosphate molecule is attaching to it, which is attaching to its third carbon number 3. And the phosphate that was attaching to the carbon number 1 is now attaching to the carbon of the ADP and converted it into the ATP. So 1,3-bisphosphoglycerate has now converted into 3-phosphoglycerate. Now this 3-phosphoglycerate is converted into 2-phosphoglycerate. Means now there is only the change of the position of the carbon, the position of the phosphate group. Means this phosphate is attaching to the carbon number 3. This phosphate will now attach to the carbon number 2 and change this molecule into the 2-phosphoglycerate. No other conversion of anything takes place here. All the other structure is same. Here is the COO is attaching, CHOH is same, and then CH2O, CH2O. All the other structure is same. Just there is the transfer of the phosphate group from the third carbon atom to the second carbon atom by the use of an enzyme, that is phosphoglycerate mutase. In the ninth step, this 2-phosphoglycerate molecule has now converted into phosphoenol pyruvate, and the phosphoenol pyruvate, how it is formed, that one hydrogen atom from the carbon number 2 and the one OH from the carbon number 3 is combined to form water, and that water is released from the step, and here there is a formation of double bond in the phosphoenol pyruvate by the use of enzyme, that is enolase. Means in this step the water is released from the process of glycolysis. Now this phosphoenol pyruvate in the last step is converting into our product, that is pyruvate. How it will convert it into pyruvate? Let's see. That the phosphoenol pyruvate, you are seeing that it is have a phosphate group attaching to its second carbon. It will break down, and then this phosphate will attach to this ADP molecule, converting it into ATP molecule. And now this molecule is devoid of any phosphate molecule, and you are seeing that if we remove a phosphate molecule from the phosphoenol pyruvate, it will result into a compound that is called pyruvate. Pyruvate have the 3 carbon atoms, 3 hydrogen atoms and 3 oxygen atoms with the 2 double bonds. So this is the last step of the glycolysis process, and you are seeing that there is also a formation of ATP molecule here. Means in the last step in the previous step in which the ATP was formed, here in the 7th step ATP was formed, and now in the 10th step ATP is formed. So there will be the formation of 2 ATPs in the 7th step and 2 ATPs in the 10th step, because the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are both converting into these compounds. So there will be the total formation of ATP that are produced are 4. Previously, we have seen that the ATP used were 2. One ATP was used in the first step and the second ATP was in the third step. Now the ATP produced, total number of ATP produced are 4. 2 in the 7th step and the 2 in the last step. So if we talk about the net production of ATP, there will be the 2 ATPs and that are produced in net amount, because 4 are produced, out of which the 2 are used. So the remaining production of total number of ATP are only 2. I hope you all learned about the glycolysis process. If you have any confusion, you can comment in the comment section below. And if you like this video, share this video with your friends. Thank you so much.